those. Can
You
you
see mommy glasses? Can you see my glasses? Okay, I see the chapter one.
Okay, so let's see. Where are we at? 11:32. I think maybe we'll click through just the outcomes for each chapter. And then if there's anything that you want me to stop on and go over in more detail, then yeah, holler. How's that?
Okay. Okay, sounds good.
All right, so the first chapter, it was sort of a general differentiate among bacteria, archaea, and eukaryotic microorganisms. And that's really in general, and then we went through that in more detail in chapters 3 and 4. So are we good on that?
Yeah, I am.
Okay. How about a fourth type of microorganism?
Would that be the virus?
Yep, that would be the virus. It's a fourth one that doesn't fall into any of those categories. And then if we went relative size, which would be the largest of all these?
The largest be the eukaryotic, right?
Right, and then
Yes.
Then bacteria.
yeah, these two are going to be about the same. They were considered the same until fairly recently, until we could look at them on a molecular level. So they're about the same size.
The virus is the smallest one.
And our viruses are the tiny, teeny ones that we're all getting a close-up view of in the media all the time with coronavirus.
Yeah.
I know.
Let's see. So I don't want to go through every one of these. I just wanted to hit for chapter 1 the learning outcomes and make sure we're good. Okay, so the timeline, there's not a whole lot of the history that we need to do, but there are some key players. So I sent the Google Doodle, the information on that Google Doodle. I don't think that Semmelweis is in our book that we have now, but he's usually in the textbooks, or at least as a sidebar, because at one point that was revolutionary. He was the one who said, hey, look, if we wash our hands,
Yeah.
we stop the spread of disease, which imagine that's revolutionary. And then Louis Pasteur with pasteurization.
Yes.
And he was also involved in a lot of surgical techniques and saying, wash your hands.
And he's the one of the swan flock. Okay.
Yeah, the flask, the swan.
Yes.
The swan flask. So he disproved abiogenesis. In these terms, it's biogenesis. Genesis is the creation, bio of life.
Yes.
So until that experiment showed that unless you have a living organism to begin with, you're not going to get any living organisms. Abiogenesis, life evolving from some non-living thing.
Yes.
Considered things. So that's also called spontaneous generation. Abiogenesis and spontaneous generation is just life rising from nothing. Poof.
Yes.
And that swan neck flask just prevented any microbes from getting in, so nothing grew. That's the classic experiment.
I did have a question on the history part on the bottom of the study guide. When it asked who was the first to invent the microscope powerful enough to see, I got Robert Hooke.
So Robert Hooke invented the first one that was able to see cells.
Yes.
Okay.
But he was looking at eukaryotic cells. And then Antony van Leeuwenhoek,
He's the one.
he developed a microscope capable of seeing bacteria. So smaller than eukaryotic cells, or seeing Seeing smaller and with more detail.
That he could magnify object up to 300 times, right?
Yes.
And they wanted his microscope, but he was not going to sell it. He said, "No, this is mine."
He's
Yes,
the one
I
who
remember
said,
that.
nope,
Okay.
this is I made this. It's mine. Yeah. He's
Okay.
So he's considered the father of microbiology.
The father of microbiology. Okay.
Yes, but right, and Hooke didn't. He was one, and that's why we have the term cells. Was from his look at microscopic life, is because he looked at an oak tree, at the cells in the oak tree of the bark, and what it looked like were cells in a prison, just little squares, or in a monastery with the monks lived in were cells, where they were all the same size, repeating square units, and so he called them cells, and that's how we got that term applied to it.
So whenever on the test, like they asking who is the okay, so for the microscope we know is the Lewin work.
Yes.
Pasteur was the pasteurization, right?
Right.
And he was the one that he believed that living things come only from other living things, basically.
Correct.
And that's when the biogenesis hypothesis took place, right?
Yes, correct.
Okay. Because they believed in, before that, in spontaneous generation, which things just came up all of a sudden from nothing. Okay. Just to understand, I mean, this is the easy part of the test. So
yeah, just remembering the names, just because you read these names, there aren't a whole lot of questions on them, but some will keep coming up. The other one that comes up is Lister.
Oh, Lister.
We have Listerine from
Listerine. I remember that.
Disinfectants.
Yeah, the disinfectant, yes.
Yes, and then we'll talk more about Koch, about Robert Koch, K-O-C-H, actually in chapter 11 that's coming up.
Okay.
He's the one who came up with a method to demonstrate that a specific pathogen caused a disease.
Yeah.
So anthrax, right?
Yeah.
He found out that.
Well, and so he came up with, the steps that we use. And that's, they're called Koch's postulates. And we'll go into those in chapter 11. Chapter 11 is a pretty fun and interesting one. So, so we have more with him. So I don't think he comes up on the final exam, and he'll be on the next exam, but not on exam 1.
Okay.
So as far as the timeline, those cover really, I think, the names that come up on any of the exams.
Okay.
Okay, there's our swan neck model. 1.4. So nomenclature, taxonomy, classification. That's just trying to think if that would differentiate you. Nomenclature is the name. Taxonomy is how we organize them, and classification are sort of that's the subunits that taxonomy divides them into.
Okay.
This one is trying to get back to think like a high school biology lesson where all the living organisms in the world, like life, and then how do we classify it? What's the taxonomic category or classification? Is we divide it first into domains. So our domains are prokaryotes, archaea, eukaryotes, and then we can divide them from domain into kingdom. So eukaryotes can be divided into plant, animal, fungus, or protist. And then we subdivide that. So animals are then divided into chordates or invertebrates, and then we divide, that's phylum, and then class, order, family, genus, species. And there are a whole bunch of mnemonic devices to remember that order. So I don't know if you've ever heard, Dear King George came over, no, sorry, Dear King Philip came over from Germany swimming.
Is
That's
that a mnemonic for?
Yes, that's the mnemonic. So dear domain, king, kingdom, Philip, phylum,
Okay.
Philip came, class, over, order, from family, Germany, genus, swimming, species. So that's just how you can remember from the broadest to the narrowest category. Yeah, in this class, really, because bacteria divisions are really, really much more confusing and constantly changing. Domain, kingdom, genus, species are pretty much what we deal with. And then we practiced writing by name. We hyphenate. Hyphenate. We italicize. We italicize the name, capitalize the genus, not the species. I think, Elenia, you underlined yours. I was so glad you did that because I wanted to bring that up about underlining used to be a standard only because typewriters couldn't do italics.
Mm-hmm.
Now we use word processors. That last one's not going to come up on the test, but it's just interesting, the difference between traditional and molecular approaches to taxonomy. So traditional is about how we divided things initially. How did they get into the genus, the species, into the groups that they were divided into? And it was all based on physical appearance, so what we could see. And now that we have, we have ways to look specifically at DNA. A lot of the traditional taxonomy is being changed. Then we find things that we didn't know were related are more closely related than two things that might appear similar. So that's just a little interesting. That doesn't.
Makes sense. Traditional is what we can see, and molecular is more in depth, the DNA.
Right, we're looking at the
Okay.
DNA, at the proteins, at the specific molecules they're made of. Like bacteria and archaea are a good example. They look alike under the microscope, they're the same size. It's not until you get down onto the molecular level that we see, oh, their ribosomes are different, that we can get that detailed as, oh, now I can pull out a ribosome and see what it's made out of. And their ribosomes are different from the prokaryote, from the bacterial ribosomes.
Okay.
So that's how we got a new classification with archaea, their own domain. All right, let's see. Naming, classifying. We divide things up. All right, any questions, anything on number on chapter 1 that you're under? It's a pretty big overview.
I'm good.
Okay, and then we'll go into, we get into the other detail as we move on. I don't remember if I mentioned this in class, but I'll mention it now since labs are being done sort of online. We don't do chapter 2 in class because chapter 2 is all about what you're doing in lab, the things you'll be doing in lab. So as you're doing the virtual labs, you want more detail or something's not making sense, look in chapter 2. There's a section on the media that you use. There's a section on microscopes. There's a section on staining. So it's got a lot of additional information for lab to help you out. All right, so our next chapter in class is chapter 3. So this is all about bacterial structures. What are the structures that all bacteria possess?
Yes, wait, I can't remember.
Ribosomes.
The ribosome, yes.
Yes.
The ribosome, yes.
And if we think about just the things that all cells possess, would fall into that. So we have to have a plasma membrane or a cell membrane, same thing, those are synonyms. Cytoplasm, We need the aqueous solution inside the cell, ribosomes to make the proteins, and we have to have DNA. So some form of DNA, either a single circular chromosome in bacteria or thread-like chromosomes in eukaryotes, but got to have that. And then, so there are groups of bacteria that don't have a cell wall. The mycoplasmas don't have a cell wall, but we don't talk about those at all. We don't look at those. For the world of clinical purposes primarily, everybody's got a cell wall, with some very few exceptions. And it's peptidoglycan. And then 3 structures that some but not all bacteria possess.
It's the cont'd, bacilli, and spiral, right? Is that what they ask?
No.
The shapes?
So this is 3 structures that some but not all bacteria possess. So these are parts of the bacteria. So like some have flagella, but not all have flagella.
Oh, okay. Some have a peptidoglycan and some don't, right? Or
For our purposes, they all have peptidoglycan. There's only really one group, the mycoplasmas, that we don't talk about at all. That don't have, but gram-positives and gram-negatives and mycobacterium, they all have peptidoglycan. So that peptidoglycan's important. It's an important way to recognize them, and it's an important target for antibiotics and disinfectants because almost all bacteria have that. So there is the one exception of mycoplasmas that we don't really get into. So I'd agree. I'd go with peptidoglycan as all bacteria that we care about have. But things like the glycocalyx, some have a capsule, but not all. Some have flagella, not all. Some have fimbriae, not all. Some have a pilus, but not all. So a lot of those external structures.
Okay.
Here's where we get into the shapes. So our 3 major shapes, these are our bacilli, cocci, and then spirals. So our spirals, they all kind of fall into the same category, but there are vibrios that are kind of comma-shaped or C-shaped, just sort of rounded. And then we have some spirals that are flexible and some spirals that are more rigid. But they all fall into the spiral bacteria. And then how about arrangements? So I don't know if we don't have enough lab for you to get into arrangements.
Is that like bacillus and cluster and
yeah, so the bacillus and coccus are here. So cocci means round, spherical. Bacillus means round shaped. And then I can put that with a prefix or with an adjective that describes how the cells are arranged next to each other. So some cells are just single cells, but if they're joined, I can have diplo, meaning 2 of them are joined, or staphylo, meaning they're a cluster, or strepto, meaning they're in a strip, they're in a chain. So streptococcus, or streptobacilli, or staphylococcus. We don't get staphylobacilli. I can get diplobacilli, 2 bacilli put together, or diplococcus, 2 round ones put together. So those are, tetra is 4 together, but really the ones staphylo and strepto are the big ones.
Okay. The cluster is the easy ones because it's all a cluster.
Yeah, so staphylo. So when you have staph infection, that's staphylo, a cluster of cocci, staphylococcus. And one thing that it's easy to confuse is that is the genus name, Staphylococcus, but it can also just be an adjective describing the bacteria. So it doesn't have to, if there's Staphylococcus, I can have spherical cells in a cluster, So I could describe them as Staphylococcus without italicizing it or capitalizing it. And that tells me, oh, it's just describing what they look like under the microscope. But I don't know from the appearance if that's really the genus. They could be Micrococcus, looks like that. There's a whole bunch of other genuses that can look the same way that aren't that. So I can use that as just, just an adjective describing what they look like under the microscope, or I can verify, do more testing and figure out that that's actually the genus name of them, Staphylococcus. So just, it's not always the genus name. And just because they look like that doesn't mean it's that genus. And you'll see that in lab, I think, in your virtual labs. Does that make sense?
Mm-hmm.
Okay. And then this is the important, this is the key to this whole chapter right here. Different structures. So here's what all bacteria have: the membrane, the chromosome, the ribosome, and cytoplasm. And then, they've got cell wall here. I would, for our purposes, that peptidoglycan cell wall is always present. But then all of these other things, some have, but not all. So you want to make sure you're familiar with those features. Oh, there's our shapes and arrangements.
Yeah.
And here are, so diplo to strepto, a chain. Tetrad, I get 4. We don't really see those. Here we see staphylo, strepto. The next section is described function of 4 different types of bacterial appendages, how the flagellum works. This is how the flagellum works in the presence of an attractant.
It goes towards.
Yeah,
right?
an attractant, it would go towards.
Yes.
And our bacterial.
Because it sends a signal, a chemical signal, right?
Right. Yep. And it
Okay.
gets attracted toward that signal. And then that flagella is attached in between. It's actually sunk into the cell wall, and it anchors there. And that anchor is called the basal body. That anchors
Yes.
the flagella, and then the flagella spins like a propeller instead of like a whip-like motion like eukaryotic flagella. Let's see, there. So we have our hook, our basal body in between the membrane and the wall. The arrangements of the flagella. So usually if you look at the prefix, if you see trichus, it's talking about the flagella. And then the prefix can kind of mono, one flagella. Amphi, like amphibious. Amphibious, you have 2, one at both sides. Peri, around the peripheral. So peritrichus all the way around. This one, Lophotrichus, is that there's a bunch or a tuft at the end. Knowing those terms, they come up probably on the exam and definitely on the final.
There they are.
Yep, so there's mono, Lophotrichus. That's my axial filament because that's a spiral, but that would be Amphitrichus, and then Peritrichus all the way around. Chemotaxis, run toward them. All of the outer layers. So on structures, we can see the capsule. It repels the stain, so we get a clearing. Come on now. Let's see. So this is really going through the 2 main types of bacterial envelope structure that's really talking about is this, the gram-positive and gram-negative cell walls. So how do these two differ?
That one has, oh goodness, I can't remember. The one can stain and the other one can't because they have a thinner, I can't remember.
Yeah, so the
just-
staining process lets us differentiate them.
It's,
The way
I
it
know it's-
happens, so when we say gram, the gram, this word gram is just the man's name who came up with the process to differentiate them. Gram-positive cells have lots and lots and lots of layers of peptidoglycan. It's very thick and strong. It would be like putting up lots and lots and lots of layers of fencing. So it's very reinforced, very rigid. Gram-negative cell walls have peptidoglycan, but they only have 1 or 2 layers of it. So they're not quite as rigid, not as strong. But what they do have outside of the peptidoglycan cell wall is a lipopolysaccharide layer. So lipolipids and polysaccharides, sugars, it's similar to the plasma membrane. And so it gives them an added layer of protection. It kind of hides that peptidoglycan. One of the lipids in it is called lipid A. It's an endotoxin. So the reason they stain differently is when we first put the primary stain on, it's going to stain all the cells purple. But when we decolorize with alcohol, we break down that lipopolysaccharide layer. The alcohol breaks down the lipids, so it washes away the purple stain. Here, the purple stain stays inside all those layers. It gets captured in there with the mordant. That's the Graham's iodine. It kind of makes the purple swell, so it gets stuck. It gets trapped in those layers. So when we decolorize, break down that lipid outer layer, and the stain washes away. But here, that stain is stuck in the peptidoglycan. So now, after we decolorize, these cells would be clear. We wouldn't see them under the microscope because they're transparent. So we add that counter stain, the safranin, and they turn pink. So these will be purple, and these will be pink. But the reason they stain differently is because here I've got thick layers of peptidoglycan. Here I've got that lipopolysaccharide layer outside the peptidoglycan. And then here, the substance in the envelope structure of some bacteria that can cause severe symptoms in humans. That's that lipid A, the lipid that's in the gram-negative cell wall. That when the cell dies, it releases that endotoxin, and that causes all of the symptoms that we see when people are actually getting better. Let's see. Here we go. So here's this picture of, here's my gram-positive, layer, all these layers stacked together with peptidoglycan. So very thick, rigid wall. And here, my gram-negatives just have this thin, a couple layers thick of peptidoglycan. So not a whole lot. And then they have this outer membrane, though, that's really similar. They both have the inner plasma membrane. And you can see this is, hey, look, it's a lipid bilayer, just like this. It has proteins, just like this. It has some phospholipids. So it's similar to this membrane. And that's giving it some protection from phagocytosis, because our white blood cells see this and say, oh yeah, it's just plasma membrane. And our cells have plasma membrane, so it doesn't really see anything different. But here we see peptidoglycan. So that makes it harder for them to be phagocytized, for immune system to see them. But these guys are very visible. That gives them a lot of extra strength too that we're going to talk about pretty soon when we talk about growth and control of growth. And this is just showing what that structure of the peptidoglycan is. I've got peptide. Well, peptides, those are the bonds between amino acids. And then glycan, glucose, that's a sugar. So peptidoglycan, what I have is all these repeating sugars. So sugar, 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 sugar. They're called NAG and NAM. N-acetylglucosamine and acetylmuramic acid, just all NAG and NAM. Those are my repeating sugars, and they're held together with these short peptide chains, short chains of amino acids that link the sugars. So here's amino acids linking this sugar chain. So that's the basic structure of peptidoglycan in both the gram-positive and gram-negative bacteria. And there's the staining process. Acid fast, my mycobacterium have mycolic acid. These would really be, these are the ones that when you do the staining process, we have to do a different staining process because they have a waxy coating outside, mycobacterium. That waxy coating is mycolic acid. And so they would technically be gram positives, but the gram stain doesn't work on them because of that waxy coating. We went through that in class. I think we went through the structures in class. I'm hurrying through this because I want to make sure we have time on chapter 8 and on the activity 1 to go over that. The endospore, we talked about the endospore. Here's the process of it forming. Any questions on chapter 3?
In the nucleoid.
Are we good on chapter 3?
Yeah. So I think I'm getting a bad echo, and I'm wondering if those who are just listening, if we can mute our microphones at the bottom on the left, and then you can just like unmute it when you're ready to say something, because I don't know if you guys are getting a lot of echo.
Yes, yeah, that's a good idea. Thank you, Angela.
But yeah, so like on the bottom left where the microphone is, you can just click it, and I've been keeping my mouse over it that way because you hear the echo.
Yeah. Yeah, good suggestion. Yeah, so just if everybody goes on to mute, and then when you have a question, just unmute and jump in so that it's easier to hear, I think. Good suggestion. Thank you, Angela.
Oops.
All right, so chapter 4, our learning outcomes. The first is 4.3. Oh, look, we jumped right to 4.3. So the first part of chapter 4 went over eukaryotic structures, the things that we have, and the nucleus is really the big defining feature. That's the one that we see under the microscope, that it's the basis for the differentiation between the two. Membrane-bound organelles. So any of the membrane-bound organelles would be a way to differentiate because bacteria don't have organelles surrounded by a membrane. So they have ribosomes, which are an organelle, but ribosomes don't have a membrane around them. So those are the big ways, and our eukaryotes are much larger, so easier to see on average, easier to see. We don't spend a lot of time on the fungus and the parasites. There are some that are very common, and so we talk about them, we bring it up, it is part of this. But you really need a parasitology course or a mycology course to go into more of the detail on that, which those courses are available at MUSC and at College of Charleston. So So parasitology is really interesting, icky, but really very interesting. So if you are interested and want to do more, we don't go into a lot of detail on them because it takes an extra level of lab safety to work with them in the lab. And because there's so much involved in it that really we touch the surface and can't do more than that. It really is a full course. Both of them are full courses in themselves. So when we look at our fungus and our parasites, our protist and Helminth parasites, it's just really a quick touch on this because working in healthcare, probably going to see it. We talked about this in general. Heterotrophs, we get food by eating it. Saprobes are sometimes called detritivores. Fungus are saprobes. They get their nutrients by breaking down dead organic matter in the environment around them and absorbing the nutrients. And parasites have to live in a host. They can't survive outside of the host. Sometimes it's multiple hosts, as we saw with the flukes. Sometimes it's a single host. Sometimes it's a host at one stage of their life cycle, but they can also be free living or in the environment at other stages. But they're dependent on a host for at least part of their life cycle. We'll look kind of quickly at some of the structures of fungus. So got dimorphic. We talked a little bit about these. So our basic structure are these threads, these hyphae, H-Y-P-H-A-E. Sometimes you'll see that pronounced hyphae or hyphae, depending on if you're pronouncing it like Latin or American English. But the hypha are the long thread-like structures, and then when you get a whole mass of them, like in a fruiting body, they're mycelium. And then the yeasts are the single-celled version. These, we talked to somebody posted about Aspergillus, about histoplasma, fungal infections in agriculture. So, yeah, fungus is really fascinating. We did mention spores, and be really careful. Always read to make sure, because we colloquially use the term spore when we mean endospore for bacteria. Bacteria have endospores, and those are dormant structures for protection, versus fungus who use spores for reproduction. They distribute these, and then these are like seeds. They germinate into new hyphae. So asexual and sexual spore formation, we talked about that pretty quickly. Our protozoans, so zoa, animal, proto, early or first animals. These are single-celled. Let's see which ones we want to spend any time on. The trophozoite, tropho, like trophic levels in a food chain, where they eat. Zo, whenever you see that zo, it's animal. So this is the feeding animal. And our cysts are dormant stages. A lot of times this, especially with fecal oral root things, this is where we get them. The organisms that we reviewed in class are the ones that I've seen come up ever on the final exam, and that you should be able to tell, to recognize what they are on exam 1. So the flagellated Trichomonas has a flagella. Naegleria is our amoeba, our brain-eating amoeba. Plasmodium, one that causes malaria. Toxoplasma, that's the reason we don't change litter boxes when we're pregnant. Somebody had asked about what's the one that causes diarrhea that you get from swimming pools. Cryptosporidium is really common intestinal protozoan that's passed fecal oral route and common in community swimming pools in the summer. Okay. Then our helminths, the stages of a typical helminth. Like, we really don't go into that much detail other than just recognizing the helminths are large worms. The reason we look at them, these are don't necessarily fall into the microscopic world, but the way they're contracted is through eggs. So that's why we study them in micro, but we've got tapeworms, flukes, and roundworms. Not all of these are parasites. A lot are just free living. We just find them everywhere, but some are parasitic, and so those are the ones that we deal with. So our flatworms, platyhelminthes, or nematodes, can be divided into tapeworms and flukes. And then we have the roundworms. Tapeworm, long tape-like segments, and this is, each segment's going to fill up with fertilized eggs at maturity, and that's how they're passed, is through the feces. The little tape pieces fall off, and then something else picks them up and gets it. Fluke. So most of them attached to the intestinal wall with some sort of sucker attachment. Asperus. These are general worm morphology. If we don't go into a whole lot of detail, again, like I said, pinworms, trichinella. This is the one we get from eating undercooked pork. And then flukes, tapeworm, tinea is the tapeworm. That's it. We don't really should be familiar with this, but don't spend too much time on these. Yeah, there's the life cycle of pinworm. Yay. All right. So this gets us, any questions on those?
So what was the fluke? Was that just the one that had the attachment that like could go on to your intestine?
Yeah, so that one will attach, and the flukes travel. The flukes are the flatworms, and a lot of those, somebody did, I think, schistosoma in our discussion, or no, it wasn't schistosoma. It was another. A lot of these have really complex life cycles, and they're common names like the blood fluke. It penetrates the skin in its larval stage, but a lot of them have complex life cycles that require multiple hosts. So some have to specifically go through the intestinal tract of a snail, then they're released and they're picked up by a fish, and then they're eaten by humans. They have really, really complex life cycles, and depending on where they end up, once they enter the human, they usually travel through the circulatory system until they get to the specific part where they get their common name. So you'll have a lung fluke, a liver fluke, the organ that they primarily affect is where they get their name from. But yeah, they're flatworms, and they attach by means of a sucker at the oral end. And then
Thank you.
then they stay in the humans, and it's really, so that's, we don't talk about this a lot, but I think the book does mention is the thing with infections with any of these parasites is what makes them harmful is the numbers. So pretty much if you've eaten pork, you probably have had one of these parasites or worms. If you've eaten meat, if you've eaten fish, we all get infected with these, and it just doesn't, it's not a big deal. what makes it a big deal is the load. So if you continuously are exposed to these and you have a high load, then we get sick. But having one or two. So there's a lot of research right now into autoimmune disorders. This is just an interesting aside, that we have this long evolutionary history where we have been, basically living with parasites in us, with worms in us. And it's not until maybe the last 40 to 50 years that we have become so separated from the environment that we no longer, as a normal course of life, have worms infecting us. And that decline, or that increase in cleanliness, the decline of worms, has also correlated with the increase in autoimmune disorders. And there's a lot of research going on to things where, hey, you know what, if you infect people with some of these parasites that we try so hard to get rid of, their autoimmune issues go away. So there's a hypothesis that our immune systems are so evolved to look for and fight off these eukaryotes that when we don't have them, the immune system is still looking for something to fight off, and that's the basis of a lot of autoimmune disorders. It's just interesting, kind of wild, but sort of makes sense that we have a couple million of years of being infected with worms and just a few decades of not having them present. So that's an interesting little aside there. Anything else on eukaryotes? All right, so for Chapter 8, I did post those 2 video lectures. Do we want to go through anything in Chapter 8? Any questions, or do we want to go to the activity?
Well, I have a question for Chapter 8.
Okay.
So I totally did wrong my activity for Chapter 8 activity, the genetic, everything is wrong. Yeah. And now we have the codon mRNA is how that's how we have the amino acid, right?
Correct.
Okay. The tRNA, they are the anticodon.
Correct.
Right? Okay. So when we have to find amino acid,
when we have messenger RNA, that's our codon. Okay.
Yes.
And that's when we go to the codon table.
Okay.
So I gave you the anticodon in that puzzle, just so you could see the complementary base pairing. And really, if you think about, if here's my mRNA, and I have codon, 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 the tRNA is just out here floating around, and it doesn't know, each tRNA has a specific amino acid that it can carry, but it doesn't know what order they go in. So the tRNA doesn't know what to do with its amino acid. What the anticodon does is, because it's specific, one codon has one anticodon, just the complementary base pairs that fit, that lets the tRNA put that land on the right spot and put its amino acid there.
Okay.
The anticodon doesn't match up anywhere else on the mRNA, so that's the only place it can put it. So that anticodon ensures that the amino acids are put in the right order based on what the messenger RNA says. So if you think about a sentence and there's a specific word order, the messenger RNA tells you what that word order is. The only thing the tRNA knows is, this is the word, this is it, I have this word, I have this word, but they don't know what order to put them in. So the messenger RNA is what makes sense of everything. That's my important.
Okay, so because what I did, I followed the tRNA to find the amino acid. That's why.
Yeah.
And then yeah.
That's a common mistake. So just remember, it's anti.
Yeah.
Anti means not.
Yes,
Anti,
and then I realized that I'm like, no, I did wrong. I'm like, no.
That's a pretty common mistake.
But it's okay because at least now I know for the exam what to
Yep.
look for.
Right.
Yes.
Once you make that mistake, then you say, oh, okay,
Now I'm
the right way.
yes. So it's okay.
Oh, let's
I'm not sure if you can hear me, but I have a question on number 10 for the worksheet as well.
Yes.
Okay, number 10, I had a really difficult time. I ended up getting the answer, but I'm not exactly sure how I got the answer. So if you could kind of explain number 10 a little bit.
Yeah. Okay. Let's these are the three questions I think people struggle with the most on here. So the first one is replication. And I think here's what you were given. And everybody defines semi-conservative replication correctly. And then if you just go back to the question, people want to make this hard. People want to make it so hard, but replication is just making a copy. So I start with this. After replication, my results are, hey, look, two identical molecules. This is exactly the same as this, and this is exactly the same. But what semi-conservative replication does is, here's my original DNA. It's a double-stranded molecule. Helicase comes in and separates out the strands. So there's one, there's the other, separated. And then DNA polymerase comes in and uses complementary base pairing to say, oh, here's my original. What's the complement? A goes with T, A goes with T, G. And it starts building by adding these correct bases. And it uses the other side for a template as well and adds these bases. So what I end up with is two exactly identical strands, each one having one of the parent or original strands that was used as a template, one strand that's newly synthesized. So that was number one. And that everybody wants to make that hard. Everybody wants to jump to transcription translation. But that's all that one was asking for is, okay, copy. So 9, and then we're going to get to 10 after this and just go in order. So number 9 was complementary base pairing. And there's a reason I give you this, because when we get to viruses, we always take our template strand of DNA, build our RNA, and get our amino acids from that, the ribosome. And the tRNA just bring amino acids in. We never would take tRNA to make mRNA or mRNA to make tRNA. We don't make RNA from RNA. We never make DNA from RNA. But viruses can do that. So this is sort of introducing us to this idea that we have viruses. We're not going to call it mRNA or tRNA. We're going to call it the plus strand of RNA or the minus strand of RNA. They can come in with either one of those, and they can make proteins. They can make either of these strands of DNA or both. So they do this puzzle. Viruses actually do this. So we'll get to that in the next chapter. But so for here, all we needed to do was, this is to show complementary base pairing. Lets us solve this. I can figure out any one of these. As long as I know one, I can figure out any of the other strands. So here I would start, I have ATG, so I know my complementary strand. It's just the complementary base pairs. This is my template strand, so this is what I'm going to build my messenger from using complementary base pairing. So T is A. A is going to be U now because I don't have thymine, and C. Once I have my mRNA, I know my anticodon, even though I don't need it. But then I'd go back and use my codon to give me my amino acid. Here, I can figure out my complementary DNA, so complementary, and then my messenger RNA. I don't need that, but I can figure it out. And then CGG, I come over here, I find CGG. Here I have my codon already, so I can just jump in. And then I can go back and forth to figure out the complements. Yeah, you just had to line up the lines. So I had the anticodon. So here I can go and find my codon. It's just a complement. And then I get my amino acid. And then I can just figure out the rest of them. So did everybody end up figuring that out?
Yes.
Okay. The most common mistakes are forgetting to put a U here or forgetting to complement. So going U, oh, the anticodon is U. No, it's not. It's the opposite. It's the complement. And then reading the table. All right, so number 10. Here's my number 10. This is my normal hemoglobin. Here's my sickle cell hemoglobin. Remember, I'm just going to say I'm getting a lot of feedback and echoes here. So if you have your microphone open, can you mute that, please? Sorry, I was getting lots of feedback. So here, if I look, my polypeptide, I've got the same chain of amino acids until here. And then I have glutamate, but here in my sickle cell, it's been substituted. I have the valine amino acid now. So what I want to do to find out the minimum change, well, to change an amino acid, I have to change my codon. So if I look glutamate, where are all the places I can find GLU? There it is. I've got two possible codons for GLU, GAA or GAG. We have that wobble room, the code is redundant. So I have multiple codons that give me the same thing. This is some protection from mutation, because there's a chance that even with a mutation, I might still get the right amino acid. So a silent mutation, no error. In this case, though, to get valine, I look and see where's all my valine. Oh, there. Get valine all these different ways. GUU, GUC, GUA, GUG. Well, if I compare all of those, how I can get valine, I don't even have to know which one of these I had. It doesn't matter. All I needed to do to get valine was end up with GUA or GUG. So the minimum change needed was to have one base pair substitution. Instead of putting an A here in the second position, if a U got stuck in there, that's my mutation, change in my base pairing. So if I could work this back, this just asked for the change in mRNA. That's my minimum change in the messenger RNA needed to end up with sickle cell anemia. One base pair. This only asked for the mRNA, for the codon change, but I could go backwards and figure out what's the DNA. So the change to DNA, well, the complement in DNA to this would be C, TT, or CTC. Here, I would have CAT or CAC. So my DNA would just be that one base pair change, a T to an A. So that's what that was asking for, though, was the codon. What's the minimum change to the codon? And that's it right there. The second base needs to change.
So it was really just a matter of using the chart on that question.
Yep. So you just had to go backwards, use the chart in reverse, find your amino acid, find your codon, compare your codon.
Okay, that makes more sense now. Thank you.
You're welcome. Anybody else have any questions on our worksheet? This is probably the other area because it's slightly different. The image is different. I've got some differences from what was exactly shown in the book. But did everybody figure out where the parts are? In the book, it has the regulatory gene, my regulator, separate, And a lot of times the regulatory gene that's producing the repressor is going to be off somewhere else, right? So it comes before the gene. But just remember when you're looking at this is DNA. So here they're showing it's double-stranded, my double helix, but they just straightened it out here to show the parts. So I have a regulatory gene. A gene codes for a protein here. So if I'm going to build a protein, I have to first transcribe to a messenger RNA and then translate it to a protein. So here's my protein, my repressor protein. And if you look at this, you see, oh, look, that would fit right here. So this is my operator, my on-off switch, where the repressor actually latches on. This is what makes the repressor change its shape and fall off. So that's my inducer. That would be my lactose. My messenger RNA attaches at the promoter. So this is the promoter here, because that sort of flags the, or not my messenger RNA, sorry, my RNA polymerase. This is the promoter flags the RNA polymerase and says, hey, come on over here. This is where I want you to start transcription. And then whether or not it can proceed depends on if my regulator is blocking its way or not. So once the inducer is present, falls off. And now RNA polymerase, the enzyme that builds the polymer RNA, can chug along through these genes. These are my structural or my operon genes that build what I want. And here I want the proteins that I need, the enzymes that I need to break down the sugar lactose. And it takes multiple steps to break it down, so there are multiple proteins, multiple enzymes involved. Each step gets its own protein, its own enzyme. So here I transcribe, there's my RNA polymerase, it just chugs down the line and produced that messenger RNA, which will then go ahead and produce the enzymes needed to break down lactose. Everybody good on the lac operon? And why we need operons? Why the bacteria use them? Okay, so our cells, eukaryotic cells, don't have an operon system like this, but we have some other ways that we regulate what genes get expressed, how they get expressed, and when, because it's important for all organisms for energy conservation. That's what we do. Only produce what we need when we need it, so that we're efficient. All right, any other questions? These are kind of straightforward. All right, if there are no other questions, then I think that's it. This is being recorded, so I will share the link to this in D2L. So if you want to go back and listen to anything again, or for anybody who couldn't make it, will be able to see it, I'll put that up there as soon as I get that link. All right, thank you all. We'll schedule more of these, probably at least one a week.
Thank you.
Thank you.
Thank you. Oh, now I have to learn how to get out of here.